with that, I'm going to transition to our first element of the program. This is the keynote panel, and it'll be moderated by Dr. Hemai Parthasarathy. She earned her PhD in Brain and Cognitive Sciences from MIT and has previously served as a North American editor for the journal Nature and was a founding editor of PLOS, the Public Library of Science. And she is presently the scientific director of the Breakout Labs in the Thiel Foundation, which has been supporting early-stage companies in areas ranging from food science, biomedicine, and clean energy via philanthropic means. So with that, I'm going to turn the stage over to Hemai, and she's going to introduce the panel.
Good morning, everyone. It's a real pleasure to be here, see so many familiar faces. And it's an honor for me to be a part of this panel of very accomplished and distinguished neuroscientists interested in and working actively in the field of circuits, ranging from basic research all the way up to clinical studies. So the format for the panel will basically be 35 minutes of what I hope is a interesting discussion around the value of neural circuits in therapeutic potential. The panel we'll have 35 minutes of discussion, and then there'll be 10 minutes for Q&A from the audience. I'd like to start by introducing the panel. First, we have Anirvan Ghosh, who is the Chief Scientific Officer of E-Scape Bio, which is a company developing therapies for AD and neurodegeneration. Anirvan, I think, made this move relatively recently, because I think I just saw him last year in Basel, where he was the VP and global head of research at Roche in neuroscience. And prior to that, he was an academic scientist at Johns Hopkins and at UCSD. He currently still has an active research lab that is virtual, I'm told, across Basel and distributed across Basel and San Diego. Next, we have Cori Bargmann, who is the Torsten Wiesel Professor at Rockefeller University. Also an HHMI investigator, a Kavli Institute director, and now the new president of the Chan Zuckerberg Science Initiative. In 2013, she co-chaired the working group that developed the scientific vision for the BRAIN Initiative. Oh, and I should mention that I'm leaving out all awards, the numerous awards that all of the panelists have amassed at this point for the sake of time. Next, we have Guoping Feng, who is the Poitras Professor of Neuroscience at the McGovern Institute, Brain and Cognitive Sciences at MIT, which is my alma mater as well, although we didn't overlap. He is also the Director of Model Systems and Neuroscience at the Stanley Center for Psychiatric Disease at the Broad Institute of Harvard and MIT. And he is, I believe, a scientific advisor for Inscopics. Last, we have Tim Denison, who is the Vice President of Research and Core Technologies for Implantables R&D in the Restorative Therapy Group at Medtronic. He's also an assistant professor at Brown University. So I thought it would be helpful to get a sense or get a little bit from each of the panelists first about their research that relates to their perspectives on circuits and neural disease. So why don't we start with Anirvan?
Thanks, Hemai. I don't know so much about my own research, but maybe a perspective. And today we'll hear a lot about circuits and brain function. But I would like to take, we have a couple minutes here, right? To talk a little bit about what I think is going to be really important in the development of new therapies and of disease understanding, and that being the impact that genetics is going to have, and the importance of understanding the relationship between genetics and circuit function. So for all of us, I think the biggest determinant of our health is our genome. And we are rapidly approaching an era where all of us will have access to our genomes. Many of you have 23andMe profiles. And there's been great progress, and will continue to be great progress, in understanding the risk that genetic variation poses. And I'm going to use psychiatry and autism as an example about kind of where we are and I think where we need to be. So one of the most exciting developments in my mind in the last 5 years has been around the understanding of the genetic basis of psychiatric and neurodevelopmental illness. And we now know that these disorders are driven by variations in a number of different genes. There are common variants that are very rare with bigger effect sizes. There are there are common variants that have small effect sizes and rare variants that have very high effect sizes. And there have been very impressive large-scale studies that have identified genes that confer significant risk. The PGC Consortium study from a couple of years ago comes to mind as a great example of this, where they reported the identification of 108 loci that are associated with schizophrenia risk, a study of about 40,000 schizophrenics, about 100,000 controls. And that led to the identification of a large number of genes that are conferring disease risk. Now, that is a great starting point, and I think that that should enable the understanding of the disease. But then when you ask, well, how has that really impacted our understanding of that disease, I think there's a huge gap. And what I see often is that we're in an era where we're getting better about asking about the relationship between cells and circuits and behavior, and I think that is something we'll hear about more today. That's dominated the pages of Science and Nature and Neuron. And then on the other hand, we have the genetic discoveries, which are very exciting, which are also equally celebrated. But if I ask how much do we understand about the relationship between those genetic variations and their impact on circuit variation, I think the answer is virtually none. And so I would encourage us to think about how we bridge that gap, because I think understanding the relationship between that genetic variation, its impact on circuit and on disease, will have a huge impact on how we can progress towards developing new drugs and methods. I think I'll stop there.
Great. Cori?
So I'm going to say a few words about the importance of taking comparative approaches to studying brains. So I think we all really care about the human brain, but and while we don't want to be guilty of anthropomorphism, I think it's fair to take an approach that I think of as zoomorphism, a word coined by Jacques Pansep, to express the idea that the complex abilities and behaviors of humans evolved from simpler animal origins, and sometimes we can really understand them better by looking at other brains, and especially by comparing principles that emerge from studying different kinds of brains and seeing what sorts of ideas emerge. And I'd like to take an idea from my own lab's work on the tiny nematode worm Caenorhabditis elegans to express that. We work on the way that worms behave. We work on innately motivated natural behaviors, and there's certain kinds of behaviors that worms show that have some similarities to what you might think of as motivated or emotional behaviors in worms. They're behavioral states. Sometimes they're very active, sometimes they're very quiet. They do different things according to their own sort of internal priorities that are to some degree independent of the environment they're in. And in studying those, we find molecular hallmarks that suggest that they do have some similarities with the motivational and emotional states of higher animals and humans. For example, one of the ones we study the most, one of the major controllers is serotonin, which obviously we know is an important player in human motivational and cognitive regulation. And a counteracting one is something called pigment dispersing factor, or PDF, which is related to the circadian activity regulation pathway that's used in mammals as well. So you can find molecular relationships that say, there really is something to this idea that behavioral states have an underlying logic and a kind of molecular control that helps you to think about them across different systems and might enable you to focus on this part rather than a different part of all the different things you could study in the nervous system. And most recently, the work that we've been doing, particularly on integrating social information into these innate behaviors, has pointed us towards something that I think is extremely relevant in thinking about human disease, especially as I go into looking at some of the things, the neglected areas of human psychiatric and brain disorders that we don't think about so much anymore. One of the things that emerges from our work over and over again is the importance of physiology in regulating behavior and in neural states. That you can't just study the brain absent the rest of the organism. And a lot of what regulates these motivated states turns out to be the animal monitoring the activity of different internal systems and modifying its behavior accordingly. Over slow time scales, often transcriptional or endocrine, that might last over hours or longer. And thinking about that reminds me that the cases where we really understand what has brought on, for example, depression in humans, tend to be endocrine and physiological, not cognitive. So thyroid hormone deficiency is one of the strongest, has an incredible effect on inducing depression. We don't really know why. We don't really know what brain circuits are being affected by thyroid hormone. Postpartum depression has just been sort of explicitly recognized as an important human psychiatric condition. That's triggered by the giant hormonal changes. Again, transcriptional changes, physiological changes that are being integrated into the brain. Action of the immune system. Before the new hepatitis drugs came out, when interferon alpha was the main way that you treated hepatitis C, those people had to be put on suicide watch because their risk of depression was so high. And I think that this is an example of something that we haven't been focusing on that much in neuroscience and in psychiatry, and that maybe the connections that you find looking between a worm and a human could help us to think about some of those areas more broadly. So circuits are not just two neurons connected. Circuits are the whole body.
So I want to, I think these are great points so mentioned. I want to emphasize just one point. I think also, in my view, is critical for our understanding the circuit mechanism of behavior and the disorders, but also for developers treatment. That is, as Animal mentioned, we have now understand a lot of genetics of the psychiatric disorders. But most of the studies so far, if you go look at it, we made a lot of progress in understanding whether it's autism or OCD or other psychiatric disorders. Most of the studies are actually use animal models in adult state. And we all know that these genetic factors play a role very early. For example, Matt State studies show that many of the genes related to autism and schizophrenia, actually the highest expression is during embryonic development in humans. So there are two main reasons actually it's critical to study the early development stage of the circuits, because although we know the adult brain is very plastic in some region, but imagine that during development, our brains are extremely plastic. Any small changes in activity could lead to a huge difference in circuits assembly and the permanent change of the connectivity. So without understanding that process, although at the later stage you will see many different phenomena or use this fantastic microscope, but you see many different changes. But you don't know is which one is actually the causes and which is the consequences, because the brain has an incredible ability to compensate for loss of the function in different neurons. So if we really want to understand or develop biomarkers, we need to go back to look at very early on what is the initial insult to the circuits, to the brain from genetics or environment or some other physiological effect, how that can actually affect the circuit development. Without that, we are going to chasing a lot of phenomena at the circuit level may not be critical to the development and the cause of disease. So by looking at it much earlier, and that will allow us not only to intervene much earlier and can correct the problem, but also to actually, if you look at it earlier with some of the studies already published from, for example, Bernardo Sammartini's lab and other labs, the early defect may be much simpler to actually compare to later on, because the compensation mechanisms have now been kicked in. That will give us a much clearer idea how to develop a treatment and how to actually develop biomarkers to trace this kind of disorders. I think once the abnormal circuit is formed, to correct that is much more difficult. So this is a two-sided sword of the problem. Yes, they are much more plastic during early days, so that means any genetic or environmental insult which will lead to much bigger effect on the defect, but also give us an opportunity. Much earlier intervention may give us a much better chance actually to correct the problem and have a much better effect, much more effective to develop a treatment for this.
So I'm an engineer electronics by training, so circuits kind of runs in my DNA. But thinking about it within the neuromodulation context where I come from, I think we're at a very exciting time. When we think about systems, especially in the CNS, the deep brain stimulators for treating Parkinson's disease, tremor, and the like, the first generation of devices were informed by extensions of stereotactic neurosurgery, where you'd go in and use RF ablation in a specific target. And pioneers like Benabid asked the question, what happens if I provide stimulation patterns instead of an ablation? And found interesting clinical outcomes that were then, of course, transferred out of the clinic and into the commercial setting. But the first generation devices based on that ablation technique really were simplistic in the patterns of stimulation that were given. Very simple tonic, fixed rates, but also very much like the first generation of cardiac pacemakers. And so they did provide a clinical outcome. It started an industry and started innovations. But I think we're now at a cusp of getting into generation 2, where we get beyond the tonic stimulation and start to think about dynamics, especially putting the dynamic of phys and physiology into devices themselves. The challenge is that sometimes in industry we can bring a bias, and so moving from a cardiac system, we went from tonic to dynamics, and in some cases we've applied similar principles in neuromodulation and with fairly mixed results. And so realizing that, kind of my team and others were stepping back and saying, before we make this leap into the next generation of tools, we need to next generation of systems, we first need to build the tools to help bridge that gap. And so thinking from a circuits perspective, if you walk into any electronics lab, you'll always see an oscilloscope on the bench. And if you asked an electronics engineer to debug a circuit and you gave them no instruments, they would look at you askance. And so that's kind of where we're at now, is trying to put basic instrumentation inside of devices that helps us to inform, as someone said earlier, both the nature of a quote, and I put this in quotes, a more normative neural circuit, what happens during disease, especially when clinical symptoms are presented, and then working with clinician researchers, understand how we might provide stimulation or other therapeutic interventions to actually bring that physiological state back to a more normal process. So that's where we're at, and that's where I'll talk about is how we need to build these tools to help bridge the gap between the generation 1 of devices and the generation 2.
Great, thanks. So you talked, Anirvan, about the tremendous progress we've made in the genetics of disease, and you are a sort of molecular therapeutics, certainly one hat, molecular therapeutics. Could you expand a little bit on really why it is so important and helpful to understand the circuits when we might just go from the genes to a therapeutic hypothesis and go from there?
Yeah, so I think, it kind of relies on this assumption that the behaviors that we see that are affected in these disorders are because of effects on certain conserved circuits. So that's the way I'm starting. So the other side of it is that if you look at a disorder like autism or schizophrenia, it is defined by certain core behavioral domains, right? And we believe that that is because of dysfunction in certain kind of specific circuits. Now, the reason I think it is important to understand the relationship between the genetic variation and the circuits is that eventually I think that the interventions that might be broadly effective have to be effective at that level where it is correcting the circuit dysfunction independent of what the original genetic hit might have been, right? So in a monogenetic disorder, I can say that it might be sufficient just to target the disorder. Huntington's disease, you figure out a way of how to target Huntington protein, and I think that probably should be the approach. When you take a polygenic approach, I don't see that as being viable in terms of a therapeutic approach, saying that, okay, you have 5 genetic risk factors with different levels of penetrance, how are we going to target that? So you have to find a point of convergence, which is going to be a point of attack. And I think that you're going to learn by understanding the relationship of these genetic variations to cellular variation in circuits, so that you get close to that point where you say, oh, if I intervene over here, then I'm likely to affect the disease for people that might have had a whole slew of different kind of genetic risk factors that drove them to that state. So that would be my reason to think that it's important to understand that relationship.
Mm-hmm. Guoping, I think you actually are studying this link. Can you sort of talk a little bit more about that, or?
Yeah, I think this is a point out really well. With the massive genetic data now, we all understand even for the same gene, right? Exactly same mutation, their clinical output might be very different. So, and that means that so it's unlikely that the autism has one actually common mechanism for all the symptoms. So most likely the different genetic factors will converge on different circuit level to produce this different aspect of behavior. So understanding, in my view, understanding not only what are the circuit output for individual symptoms is critical, but also subgroup of different patients into subgroups. In my view, in the future treatment for all different, many of the different psychiatric disorders will be subgroups of convergent mechanism. We're not going to find one mechanism. Actually, there will be many, many subgroups of them, maybe 20, maybe 100 of them. So understanding what is their converging point for the behavior mechanism is, I think, at the circuit level will be critical, because these are all, any behaviors are controlled by a circuit or a group of circuits output.
Yeah, just sort of to follow up on that idea, in the human genetics right now, certain of the risk factors that give rise to a high frequency of autism also give rise to a high frequency of infant cardiovascular disease. And these include some of the chromatin remodeling factors and even some of the same alleles. So I don't think developing a therapeutic hypothesis around chromatin remodeling factors is going to be a fruitful approach to autism in particular. I think this reflects the fact that these molecules there are only 25,000 genes, and there's way more than 25,000 things to do, and so the genes are being used in many different settings and configurations. Nonetheless, you can use them as a wedge to drive into the underlying biology. Each of these molecules has to be acting in some number of cells to have the phenotype that it has, and those cells are connected in a function, which is what the circuit does. So we can use the molecules to come in, but it's not necessarily going to be true that we can target those molecules and expect anything sensible to come out because of their intrinsic multifunctionality.
Yeah, and that's actually an area that we're curious about as well, in terms of the genetic, but also then the physiological representation. So when we talk a lot about precision medicine these days, a lot of times I think there's a bias, or just people go immediately to a genetic test and the like. And one of the areas we're curious about is the individual physiological marker. And so what we're finding now, with the researchers doing devices that can do this chronic sensing across large patient cohorts, is that there's a general representation of a physiological state and disease, but yet there's a personalized signature to it as well. And so given the complexities of treating these patients, these consumers of the technology, one hopes that in the future with algorithms and the like, and actually starting to share these different states and best treatment outcomes for different patient cohorts, precision medicine can actually apply this physiology represented in the network as well as the genetic information that's associated with the patient.
So given that we agree that this sort of circuit level understanding is going to be important for developing therapies, treating disease, and so on, where are we on that spectrum of understanding towards therapeutic intervention? It's clear, Tim, you guys are already putting devices into patients, and I think also trying to understand what those devices are doing. So there is a level at which we are able to intervene. What limits our ability so far in the kinds of, maybe in different diseases, in order to intervene?
Well, I think one of the elements is to really understand the physiological dynamics, both of the disease, but then also how stimulation or other therapeutics are coming into play. So one area where I think it's important that we actually have the tools, so in the spirit of this conference, we must have the tools, and the visibility to the dynamics is an example that I saw from Phil Starr's team at the University of California, San Francisco. And there are two elements. One is when we talk neural circuits, they're studying about cortical circuits and how they interact with basal ganglia circuits in the Parkinson's disease state. And subjects sometimes go dyskinetic. So for Parkinson's, you go from a lack of motion to basically excessive, more uncontrolled motion. And having this oscilloscope basically in the device, Phil could see a new rhythm that appeared within the network. It was not the classical brain-machine interface, gamma kind of controller. It was literally this, like almost a ringtone that arose out of the network and showed the cortical and basal ganglia circuits kind of locking in together into this state. And then what's fascinating, this is why we have to understand it, is sometimes subjects have stimulation-induced dyskinesias. And what they discovered is that when the subjects are entering this state, basically the network locks to a harmonic of the stimulation pattern. And so without this visibility, we would never have access to what's happening dynamically. And so why this is important is once we understand this characteristic of the neural circuit, then engineers like me, control system theorists, we say, we can build algorithms that detect that state, and then we can actually adjust stimulation appropriately to try to optimize the system. And that's just one example that I think is kind of fascinating, that really no one had a clue about what was going on. And then once we built the tools and get this idea, we have a better understanding what's happening. But then most importantly, there's a pathway to get this in the future out of an investigational study and into more of a real live clinical application.
So maybe I can elaborate a little bit about how, and it's an example of how this kind of circuit-based approach is being integrated into drug discovery. And a program I'll talk about is one that we worked at Roche, along with Luca Santorelli, who's sitting there when we were both in neuroscience there. And this had to do with an autism program and around modulators of the V1A receptor. And so on one hand, there was a body of literature linking kind of vasopressin signaling to social dysfunction. And but then as we started developing that program, we wanted to have greater confidence that the molecules we're developing would have effects on the kind of relevant circuits. And two of the ways in which I think it impacted that program was, one, is instead of just relying on kind of pharmacological models, animal models, we moved towards the use of these kind of genetic models related to autism risk genes that have kind of come up in the last several years. And hopefully a few years down the road, we'll have marmoset models as well. We don't have those yet, but there's rodent models to show that you had an impact on those models. But also in terms of visualizing an effect on circuit, we have a rodent fMRI set up. And so we could use that to look at differences in activity of different brain regions in kind of wild type in those genetic models of this disease. And then ask not only is there an effect of these compounds at a behavioral level, which has typically been the way it's been done, but also at the circuit level, if the circuit is defined as big brain areas, whose activities are changing, to see are we normalizing activity in those areas that we believe to be relevant for those behavioral domains. So this is just an example of how I think it is beginning to have an impact on building confidence that the molecules that we develop will likely have an impact on the relevant core circuits.
So I say as a basic scientist, I think we are, a lot of things depend on us discover the basic functional principles of circuits assembly, circuits function. I think we are at a very, very early stage right now. We even don't know in the brain how many cell types are there, and the basics how individual cell types function in each circuit. So I think there are a lot of going on right now. I think in the next few years there will be a very big advance. And I understand, like Allen Brain Institute has been doing a lot of work. I heard that Cori's new initiative will also put a lot of effort on the cell types. I think this is the critical thing. Without understanding the individual cell function, we cannot find the actually appropriate way to modulate their cell type function. Then we cannot precisely modulate circuits. I think that's only about the brain. There are a lot of, we are not talking about how the other parts of physiology regulate these circuits. I think that will be a very fundamental thing for us to understand what are cell types, what each cell types does, and how they modulate circuits function.
Yeah, I mean, just to sort of the first order understanding of the brain is to think that there's blobs for this and blobs for that. areas like the hippocampus that carry out a particular function. But the next order understanding of the brain is to realize that the important thing is that traffic flow through, that certain kinds of information be transmitted accurately and without overwhelming other streams of information that are going through the brain at the same time. And certainly deep brain stimulation is built around that principle, that by rebalancing areas of neural activity that have become that are out of whack with each other, you can actually restore function without restoring the cells that have lost their function in a disease. So the idea of circuit normalization as a powerful tool, I think, is something that is useful throughout neuroscience. I think it's also, the voice that's not at this table here is the voice of neurology. And I think neurologists have often really appreciated the importance of certain brain connections, certain pathways, damage to particular nerve pathways as really the flow of information being the step that's disrupted. And I think we can bring them into the conversation in circuit neuroscience more than we're doing now.
And at least the three of you are focused on animal models in this context. What's the advantage? Where are the strengths in these animal models? Where are the gaps between what we know in animals and what we can do in humans? How do we help to bridge the divide, if you like, between our understanding in animals and our understanding in people?
You try and get an animal to tell you that its stomach hurts. The phenotyping in human is absolutely fantastic because people will tell you what's going on, and you can ask them to do a task and then they'll just do it instead of having to train them for 2 years as you do for an animal. Where human neuroscience is in many ways extremely powerful at the phenotypic level, but at the interventionist level, it's really challenging. And so I work on the animal I work on because I can do anything that I can think of, ethically and safely, and reliably and reproducibly from organism to organism. And as you move from the very simple level of the worm to the human, those experiments become successively more difficult. But you also reach a richer and richer understanding of the more complex and dynamic aspects of behavior.
I think, Guoping, you were also talking about this bridge.
Yeah, I think, animal studies can never replace human studies, but animals are essential for us to understand. No matter what this, brain disorder we're studying, just let's say use development as an example, right? We need to know every step of what's happening in the brain. You just cannot get that information at a molecular, cellular, or circuits connectivity level. For example, neurodegenerative disorder, when you get the patient in the brain, the neurons are already dead. We want to know why they died. So we want to know every step. If we have this access to every different stage from very beginning, we know there are point mutations in APP genes, why they cause the neuron death at the end. If you can have that process to all the information from every single molecular cellular level, that probably will be critical for our understanding why at the end of a neuron die. But by the time we get the brain tissue, they're already dead. So the animal can probably tell us a lot, and you can track every step. That's, I think, is the accessibility to the brain tissue, and to at whatever the stage we need is probably a tremendous advantage to use animal. Whatever the stage, whatever the animal model we are using, that's going to be critical for us.
So Guoping, I was wondering if you could comment a little bit on primate models. I mean, I've been watching it from a distance, and I don't know exactly how deeply you're involved in this, but I know to some extent you are. And that's been something that has not been fully integrated. There haven't been ways of manipulating the genomes of primates. And I was wondering if you could just touch upon that and the impact you think that might have in the next 5 to 10 years.
Yes, so we do have a very ambitious, big program, not my labs, but from MIT and Broad Institute, and together with many, many investigators working on marmoset for many reasons. Although it's a New World monkey, but it is much smaller and group housing, very social, communicative, but also the reproduction cycle is very short, and they produce much faster compared to any other primates. And genetic modification right now is all feasible. Actually, whether it's knockout, even knocking, we have succeeded in knocking now with new technology, modified CRISPR. So all these are possible to do. And I think, in principle, there are also ethical issues. You cannot do like to C. elegans. Whenever I get hundreds of them to do, or thousands of them to do it. And we reserve these to the question we really cannot address in rodents, if we can. For example, our best example is prefrontal cortex. It's really well developed in primates, right? And rodents have much, much, much smaller. And if that's different, if we think that many of the disorder is subcortical to cortical control, for example, information flow to there, and if the prefrontal cortex is so different, then the connectivity circuit will be very different. And the purpose of developing that circuit has an evolutionary purpose for each of our behavior, is better surviving for each different species. And we survive different from rodents. So I think that all these are really feasible now. And we have been engaged with many different groups about, what are ethical things, ethical concerns, what are the technological possibilities. In about a year or at the most 2, almost everything you can manipulate in rodents, mice, you can manipulate in mice. Whether it's a big knock-in, small knock-in, knockout, precise repairment, all these become possible in mice. I think it would be very good for the community to discuss this, what's the need, what's the best use of these models. And it's very expensive, so how do we share the data, share the tissues, share all these? We intend to be completely open to anyone, whether you want a marmoset, or you want a sperm or marmoset, or you want a brain tissue, we want to make it completely open because we really want to move. We see the failure of drug development, and we cannot always blame mice. And the poor mice, they don't have lawyer to defend themselves. It's our fault because mice are great models if we use properly. So we just maybe use them not some wrong way. But so we want that, we see these suffering and pains that in development treatment, we're hoping this will be a really open resource to all the communities. So I think this is really possible now.
So it seems that we've almost down to 10 minutes left. So we welcome questions from the audience. We'll keep talking, of course, if Tom.
We're going to spend a lot of time today talking about circuits. It would be really helpful to actually explain what a circuit is. What do we mean by that?
Can you repeat the question again? I'm sorry.
What's a circuit?
I think in this setting, I think in this setting where a circuit is a collection, circuits can be many things. Circuits can be what Tim would call a circuit, which would be electrical. Circuits could be what a cell biologist would call a circuit, which might be something molecular inside the cell. I think in the setting of the brain, the circuits are defined as groups of interacting cells that communicate information between them, often not linearly and forward-directed, but through extensive feedback loops that modify their information as they go along. And from my perspective, that also communicate at a distance more slowly through mechanisms like modulators and endocrine factors to modify their function. So it's the interrelationship of cells, both physically and functionally, that define the flow of information in a circuit.
Can it be defined anatomically, or does it require a functional definition?
I would not define a circuit entirely anatomically because of my own interest in neuropeptides and neuromodulators, which are not always point-to-point synaptic molecules, but rather sort of locally acting, but not precise. But I think if you want to include them, you're going to pretty soon decide that the whole brain is a circuit. And that might be the answer that you're pointing toward, but you can divide and conquer may be at least a starting point toward getting there.
I mean, Tom, I see it, or I kind of visualize it largely kind of in terms of how we think of as electrical circuits, as cells perhaps being elements in a complex network, with this concept of output being pretty important, which is a functional output. And therefore, I think the function part, I think, in my mind, is integral to thinking about it. And the understanding, I think, is about how those elements influence maybe the activity of other elements, but eventually function. So I actually do not think that you can reasonably think about brain circuits independent of function.
So I have over here. I have a question about the importance of computational approaches. When I think about circuits, in part what I think about is the set of equations that might define the function or describe the function of a circuit. And given the approaches that are being used, including in Scopics, the opportunity to now use the data that's coming from these approaches to constrain these kinds of models, I'm curious as to your perspective on that.
Well, I'll take a crack, but I'm no expert on this. So, I think that where I see potential value is if you can describe these input-output relationships in some mathematical way so that you have an understanding of the impact of manipulating any given element on a certain output you're interested in, then I think it's hugely valuable. I don't know how far near we are, but I think if you get to that point, because we're often in this situation, and soon we'll be with, Mark mentioned, recording thousands of neurons every day. There's like, gigabytes of data floating out, and you will have lots and lots of information about activity patterns of different neurons, neuron types, they're genetically defined, all of this is going on. But then if you say, okay, where do I want to be manipulating the circuit in order to have a desired outcome? It's all very intuitive. Maybe I would like to increase or decrease, particular kind of GABA subtype. They tend to be pretty sexy, right? But I'm not sure that I really know what the impact would be of manipulating one of 10 different GABA subtype neurons. And I think there a computational or even better mathematical understanding could be quite valuable. But I would be curious to hear if there is work in this direction that's being done that you think is meaningful.
My sense is that there's relatively little that's being done in the way that you describe in terms of the development of computational approaches in the context of specific diseases. But I think it's a potentially really big opportunity to combine some of the things that we're doing in terms of data collection, large-scale data collection, in order to understand what the data mean and what kind of constraints they put on our understanding of the function of those areas and those connections in exactly the way that you described in terms of understanding the consequences of different manipulations, the ones that we might be able to make now or the ones that we might want to make, and we might want to make an investment in trying to do something very specific. I think computational approaches can provide us some kind of insight into which ones, where should we make that investment in trying to do something in a very specific way. But I think it's very much in its infancy at this point. Most approaches, modeling, computational approaches have been about understanding normal function rather than a computational understanding of the dysfunction or even repair of that dysfunction.
So since I am a neurologist, I'll take the neurologist's point of view on this as I listen, because, Tom's comment about like what is a circuit kind of gets, and the answers are varied, gets at the fundamental question of if we're trying to move toward transformative CNS therapeutics, as the title says, then the question is at what's our time frame to know enough to go to that target? And by listening to Dr. Denison, the idea is with some wild guesses and some really good luck in certain diseases, we can do things now at a very macro circuit level. How is the process for the panelists? How do you see the reverse translation? When would you be happy enough to actually have it be not perfect, but allow you to do some of these developmental, genetically viral-inspired kind of microcircuit dissections within the framework of a macro circuit that actually already is showing promise, rather than, no, we'll do it where we can control every variable and we'll eventually scale it up. So I just kind of want to throw a bomb out there now that, in fact, if we're really going to get to something therapeutic, there's going to need to be some compromises with the bidirectionality of the plan. And where are people's comfort level? Because we clinically can talk about what we need or what might be good enough, or that we can have a starting point for patients versus what will be what you have to know from the bottom up to actually move into the therapeutic platform.
I can say a few things. I'm personally very interested in translating what in the basic level on circuit level to clinical use. I think, in my view, there are a couple of things that would be critical determinant. One is, if in the adult brain, I think we could go much earlier with testing. The second is how invasive that approach is. But to me, because of previous failure in translating in many aspects from rodent model to see, I'm again say I'm not blaming the mice, and they hear me. In Chinese zodiac, I'm a mouse actually, so I'm not going to blame mice. And but that's one of the reasons that we try to develop primate models. I think if you in different models you show efficacy, it will make me much more comfortable to getting into, and I would be much eager to get into clinical to test this any of the hypotheses at the circuit of manipulations, because we are talking about circuit manipulation to try to change a particular behavior. That a conserved circuit is critical for the translation. So in my view, we cannot find a perfect solution, as Adam said, and very quickly. But if we can see a certain efficacy for a particular behavior output, then if we can try this in adult and we can confirm in different models, especially in primate model, I think that will really speed up the things. That's my view.
Cori, you wanted to add something?
Yeah, I think that there's a real difference between adult, serious, end-stage diseases for which we don't have treatments and jumping around too quickly. And I would just like to point out that I think that for human therapies, we will be doing the best thing if we do not make the kinds of mistakes that were made early on in gene therapy, which basically set the field back 20 years, or that are currently happening in a couple of ghastly cases with stem cell therapies. We want to make sure that when we move into humans, we're doing so thoughtfully and not creating a backlash against exactly what we want to do. So yes to moving what we can, especially for people who have no other alternatives, but also yes to preserving the scientific and clinical enterprises by being thoughtful.
So I'm Jim Hendricks with the Alzheimer's Association, and I would say that if we had good translational animal models today, we probably wouldn't be here in this lovely setting, because we would have solved a lot of these diseases already. So as I'm listening to the panel, I'm thinking about how important it is that we are doing this research in new animal models to help the translational research side. But I've also heard these great stories in the past of the great next thing that's going to solve this problem. Right now, I'd say in Alzheimer's, we have two categories of animal models. One is the behavioral animal models that have been very bad at predicting human disease. And we have the biomarker models that have been actually very good at translating to human biomarker research and have been very supportive of the clinical research. So forgive my ignorance with respect to the work you're doing with circuitry. Where do you see the future of what you're developing? Is it going to be more alike that you're developing new behavioral models that will predict complex diseases in humans like Alzheimer's disease? Or do you think you'll be helping to identify new markers of disease that will help to develop new therapies?
And we're over time, so if somebody wants to take a quick comment on that, maybe we'll pursue that in one of the discussions.
Yes, it is it will be very helpful developing biomarkers. We call it translatable neurophysiological biomarkers. There are actually quite a few groups, both in academic and in industry, including Biogen's. Other companies are actually, Novartis, they're all working on this, and including the Roche, your former companies are also working. I think the goal is not to develop a behavior, because animal behavior, no matter at what level, even monkeys will be very different from humans.
Okay. Well, with that, thank you all for opening up the first panel discussion for the day, and I hope this leads to many interesting discussions as the day goes on. Appreciate it.